Welcome, everyone. So nice to see you. So I would like to today talk a little bit more about the methods, more deep of what Andrea pointed to, and also talk a little bit about work in progress, what we are working right now, briefly. So what do you do? Like, I joined 5 years ago when the collection was pretty much done, and I was supposed to make the most out of those samples, help with setting up methods, processing and everything. So how do you treat those samples that's been collected by volunteers with so much effort? Of course, you treat them with respect, first of all. And we really try to find the best solutions ever. We definitely knew that we cannot sort them for the reasons Andrea told you about. This is out of question, but we need methods that are high throughput, reliable, reproducible, cost effective. And we want to know what species are in the samples and also how many individuals of each species are there. Those are the two main goals. So we knew we will do metabarcoding. Andrea already mentioned, we knew this from the beginning, right? And this is extracting DNA from a bulk sample, amplifying a little short fragment like a fingerprint of a species, and then by matching to reference databases, you identify those species. That's the principle. Okay, that's great. But what exactly are we going to do? I mean, the field is really quite new and fast evolving, and there's tons of information, and I was supposed to visit other labs, learn from other people, communicate, bring all those people together and find the best methods. But the COVID happened, so I was just sitting by myself and reading papers. And we also contributed a bit doing experiments, testing all sorts of things. And I would like to talk about all of it in detail, but we don't have time for that. But I'm gonna say that a little bit about what we came up with. So that's the overview of our FAVIS protocol that we think was the best way for us to analyze the samples. And it looks simple, but keep in mind each one of these arrows is like tons of tests, trials, failures, and it really took a lot of effort from a big group of people. So I'm very proud that we put it out there and other people can learn from us. So few steps I will show you. When we process the samples on Öland, at Station Linné, we keep each sample at their dedicated stations. The insects, as Andrea mentioned, never leave the bottle. We just decant the ethanol and process it further. So you see, each sample has its own equipment. It's very neat. We really want to avoid all troubles. And of course, every day of the processing, we also include negative controls, and we take care of all this. We measure wet weight of the sample, and to get the idea of the biomass. And Laura will not talk about this today, but she did make use of this data. It was quite interesting. We are also using this information to assign a size class to each sample. So you see colorful stickers on the bottle, and then we know how much lysis buffer we have to add, what's the best procedure for this particular sample. We add some lysis buffers, like a bath water, basically, to insects, and we incubate them in a shaking incubator for a few hours, and they just leak a little bit of DNA. Afterwards, we take the bath water out, we bring ethanol back to the sample, of course the same ethanol, and the sample goes back to the freezer house for future work. So it can be taxonomic work or molecular work. We just looked into it's just a snippet of some results about Lepidopterans how we came back to samples that had some interesting Lepidopterans in them, verified, send it to have a specialist look at it, verify that our sequences really match something interesting in the physical sample. And this is how an example of how it can proceed. Then from the bath water of the insects, we take a little bit of it, we extract DNA, we keep rest for future, let's say, work or repeats. And we purify DNA from this, and part of it we use for library preparation and sequencing, and part of it goes for long-term storage in the museum. This is like very valuable resource that we take care of. And actually, Samantha, who will talk later, she will tell you how she used some of those samples, those DNA extracts, for metagenomics work. So, but with the working stock, we proceed with library preparations that I won't go into details, but we basically, we amplify the barcodes, we match some adapters, we send it for sequencing. And what we get out of the sequencing is just a long list of sequences. We aim for about 1 million barcode reads per sample. So if you imagine a few thousand samples, every 1 million reads, it's quite many, but I will show you on an example what we do with it. So first we group them into based on similarity. So if those sequences are the same, they are probably the same, right? It's just the same sequence. That's all information we have. So we merge them, and we can also say what's the relative abundance of those different reads. And if the sequences was the same for all the reads, they call this OTU, Operational Taxonomic Unit, that Andrea mentioned. So it's a proxy for a species. Based on molecular data, we can assume that this is probably a species because it's different from this OTU and different from this OTU. And then to know what those OTUs are, we have to go to the reference library. So there's a huge database of barcode sequences. A lot of people sequenced a lot of barcodes, and they know what those barcodes are. So we basically compare our sequence to the reference library, and let's say for the first, for the blue sequence, it matches at 98 or 100%, it matches this beautiful butterfly. Oh, we had a butterfly in the sample. How wonderful. So let's look at the second sequence. What is this? Oh, this one matches this furry little guy. So we didn't even have to see him. We just sequenced the bath water, right? And he left a little bit of DNA there, and now we have him. So, and the third sequence, oh, this guy. Okay, but we know what that is, right? That's great. And the fourth sequence, there was no match in the database. So we had no match at the species level, we had no match at the genus or family level, but we know, based on the sequence just being similar to others, we know that it's some fly, it's some Diptera. It's not really uncommon. We know very little about Dipterans. So from this sample, we know there was this species, that species, and one unknown fly. Okay, perfect. So basically, using this methodology, we can say quite a lot about what species were present and what were not. And it's just a brief introduction, and Fredrik will tell you all about the difficulties and caveats in this process. So using this, we know what the species lists are in our samples, and that, how Andrea said, in Sweden we have around 33,000 species, in Madagascar 77. But now we would also like to know, of course, how many individuals of each species are there. This is relevant to understand what are they doing there and how many are there. So as everybody will tell you, as most people know, metabarcoding is good just for presence-absence data. So we can say that the sequence was there, but we cannot say how many individuals gave this sequence. But we tried to do our best, and other people are also trying to get some abundance estimations from metabarcoding data. And it's maybe not too silly to say that it's like a holy grail of metabarcoding, because if we can use this cheap method and high throughput method and get some abundances, some idea about abundances, that would be absolutely excellent. So the state of the art was that probably it's impossible, but maybe we are too naive or too hopeful, but we will try. From the literature, possibly the best way to start would be to use spike-ins. So spike-ins is just something that you know, DNA, for instance, that you add to a sample, to each sample, and then you can see it, it amplifies like a barcode, so you can see it in the results, and it can serve as a quality check. So if it amplified or not, it should amplify. If it didn't, then the whole sample is failed, and you cannot treat seriously those little sequences that you received in the sample. It can also help with normalization, so bringing all samples to the same level. If you knew exactly how much DNA, how much spike-in you added, you can like make those differences between those two samples like visible. That there was much less DNA here. So it helps in between sample comparisons. And also using superpowers of our group. I'm sorry, we hope to be able to model also abundance estimations using the spike-ins. But what type of spike-ins are there? So we use two types. Biological spike-ins, which is just a species that would not be ever caught in your malaise trap. So in Sweden, that's some exotic species that we can easily produce in large numbers, and we add them to each individual sample. And the synthetic spike-ins, there would be a DNA sequence that you can measure very precisely and add to your sample, but it just doesn't resemble anything. It's been synthesized, it's been made up. And that sounds perfect because you will never confuse it with anything. It amplifies very reliably because you always know exactly what that is. And you can add very precise quantities. So that's great, but at the time when we did this, no such spike-ins existed, so we just made them up. Just made up. Think of a sequence and synthesize it, put it on a plasmid, add to your samples, and there you go. So we also published this protocol saying exactly how to do this, hoping that maybe others will be encouraged to use it. So we test those spike-ins in a small experiment, but we also added them to a number of samples. But this is still work in progress, so I will tell you just about the experiment. So we took 15 samples, our IBA samples. We added artificial biological spike-ins right at the beginning, and we processed them with the mild lysis like we do for all samples. After the procedure, our collaborator in Singapore back then, now in Berlin, put every individual insect in a well and barcoded them. So we had barcode of each member of the sample. So we basically knew who was in there, exactly. Then we put all those individuals together again, homogenized the sample, extracted DNA from that, and metabarcoded it by adding synthetic spike-ins right before the DNA extraction. So we had two types of spike-ins, and we worked with Fredrik and Emma, who are fantastic in creating Bayesian hierarchical models. And they wrote a model that estimates DNA available for barcoding, and also amplification factor per sample. So it basically uses the information from spike-ins and trains to predict abundances for all other species present in the sample. And those are preliminary results of this. So this is basically number of specimens predicted. And this is, number of instances of predicted number of specimens minus the true value of specimens that we had in the sample. So basically, if it's at 0, that means the prediction was exactly spot on for this species. And if it's -1, then it was slightly overestimated, and if it's +1, it's like slightly overestimated. So basically, we had 451 species in our experiment. It was around 1,000 species sample like instances, and 543 of those were exactly predicted, exactly spot on. And within ±1 individual, we were actually 773 were almost right. So we did not get very good abundance estimations for species that were very numerous and very small. So basically, our biological spikings did not match how the species looks. Then it's difficult for a model to learn. But we are still working and tweaking the model, and I think we can also say that answering the second question, that spikings are useful for between sample comparisons, and the synthetic spikings help us correct for all the lab procedures we did, and biological spikings help to model the DNA coming from each individual, and some fairly good abundance estimations maybe are within reach. So it's quite a hopeful work in progress. But thank you to the whole team, and that's it.